So thanks for the opportunity to give you an update on the development of CF10. I should say at the outset that we are at the important but boring stage of drug development at the moment. Namely, we're undertaking all of the safety studies that are required before a new drug can be offered to patients. Those studies are dictated by something called the International Council for Harmonisation, the driver for which is to make sure that the studies, once performed, will be applicable worldwide, and so new medicines can be made available to patients as quickly as possible. So the studies that are ongoing at the moment, as I say, are safety and toxicology studies. And for those of you who want to escape to make a quick cup of coffee, I'm delighted to say that the studies are all proving to be very negative, namely that CF10 appears to be an extremely safe and well tolerated molecule in preclinical models. I'm not going to go into a huge amount of negative safety and toxicology data because it's really not that exciting. So what I will do though is give you an overview of what CF10 was designed to achieve, where we are at the moment, and what we'll be doing in the near future. So as I think everyone will probably know, CF10 was discovered by the late Professor Ros Anderson at the University of Sunderland. And as you heard in the introduction from Alex, Cystinosis Foundation UK very generously funded Ros's work to the tune of around £1 million that supported the discovery and initial laboratory studies with CF10. Ros was on the verge of moving to the next stage, namely the safety and toxicology studies, when tragically she was diagnosed with a very aggressive form of uterine cancer. I'd been advising Ros on her application because I'd previously chaired the MRC panel to which she was applying. And when it became clear that unfortunately Ros's tumour would not be satisfactorily treated, we agreed together that I would take over the project. And indeed, Ros heard a week before she died, while she was in the hospice at Sunderland, that the MRC had approved the funding for her project, some £1.6 million, and that in fact it was the highest rated project that the MRC panel had seen in the previous 3 years. So some consolation for Ros at the very end of her life. And myself and the CF10 team are committed, of course, most importantly, to delivering a new and more effective medicine for those living with cystinosis, but also realizing CF10 as a new medicine for cystinosis patients as a lasting memorial to Ros's research. So you will be more familiar than I with the drug cysteamine, its advantages, which are significant, but also its disadvantages. So there is no doubt, and indeed the registry data that we've just looked at reinforce the fact that it is an effective treatment for cystinosis and works best if you start taking it early. The mechanism is well understood. It removes the excess cystine that we've seen in some really beautiful microscopy and other imaging data from all tissues, and that reduces damage to those tissues and most critically the need for kidney transplant. It's a very simple and cheap oral medicine. The model at the bottom on the left-hand side that looks a bit like a dog, that's what my wife says it looks like, is a space-filling model of the drug cysteamine. There are very few drugs that are so simple, and cysteamine, or cystagon as its marketed form, is, I think, in terms of its pharmacology, a truly spectacular drug. but it has major disadvantages. The bad taste and smell, which can cause significant sickness, the irritation to the stomach and the gut, which can lead to ulceration, due to the rapid breakdown in the body by metabolism, enzymatic breakdown. Large doses and frequent doses are needed for adequate cystine depletion, with cystagon dosing every 6 hours. And this metabolism results in the conversion of the drug to volatile, gaseous, that is, compounds, which are breathed out and come out in the sweat. And because of the sulfur, as we all know from smelling rotten eggs, for example, sulfur containing compounds have an unpleasant smell. And that gives the body odor and halitosis problems that are a real issue for cystinosis patients. And a knock-on consequence of these disadvantages is that patients may not adhere to their therapy, i.e. they don't take their medicine. And in that case, because of the critical nature of cysteamine for managing the disease, there is not adequate disease control. So CF10, as designed by Ros, is shown on this side schematically. And CF10 is what we call a prodrug. It's a drug which is inactive when you take it, but it's converted within the body to the active form being, of course, cysteamine. So the structure schematic here shows in red the active component, cysteamine, in blue the targeting vector, glutamic acid, which is linked covalently, a solid link to cysteamine, to protect the sulfhydryl SH group, the sulfur-containing bit of cysteamine. In green, something called a thioester has been introduced, and then to get the drug taken up from the stomach and the gut after administration, there is in light blue an ester applied to the glutamate molecule. And the impact of the thioester is, as I say, to cover up the sulfur so that you don't get the side effects in the gut. The ester is to improve the absorption through the gut wall, and the glutamate in blue is, in dark blue, is to target the cysteamine to cells at the cell surface level of tissues throughout the body, including, of course, the kidney. And this was designed to lead to increased potency, i.e. you have to take less drug less frequently to generate lower blood circulating levels of cysteamine, the active drug. So that will reduce the conversion to the volatile compounds, reduce the bad smell, and overcome, therefore, the halitosis and body odor problems. So that was the design. What have we achieved? And this is very much the work that was done by Ros before she was unable to continue the research herself. So CF10 has minimal smell, good pharmaceutical properties, by which we mean it's extremely soluble in water and should be very easy to take and to give. In studies in mice, there was reduced or no damage at all to the gut. There was depletion of cystine in both cells grown in flasks in culture, in the tissues of cystinotic mice. So these are mice engineered to have a disease similar to clinical cystinosis, and it was active without producing any side effects in a zebrafish model of cystinosis. So this was all consistent with the design. Critically, the levels of cysteamine, the active drug in the blood, are much lower after CF10 than they are after cysteamine, and that should result in reduced production of these volatile thiols, which are so problematic. On the other side, and even better news, is that in comparison to cysteamine, you have equal or greater delivery of cysteamine into the tissues. And in particular, the kidney had very high levels of cysteamine after giving CF10. somewhere in the region of 40 to 50 times higher than when an equivalent dose of cysteamine itself was given. And so based on these results, we think that there is real potential for less frequent and lower dosing, lack of bad breath and body odor, and importantly, no damage to the gut. So those were the data that Ros generated in collaboration with groups who are represented in the video conference today. With the funding we secured from the Medical Research Council in the safety and toxicology studies that we're doing at the moment, we have seen no evidence of unwanted side effects. And so we're confident at the moment to proceed, although those studies are not yet quite complete. So what is this going to mean to those who are living with cystinosis? We believe CF10 should be palatable and easy to take. It should have reduced side effects, specifically damage to the gut, the bad breath and unpleasant body odor issues. There should be improved cysteamine delivery to tissues, and this will result in a lower risk of kidney failure and reduced need for dialysis and transplantation. We believe there should be fewer doses required each day, possibly only once or maybe twice. All of the above will give improved quality of life and greater adherence, and hence reduced risk of treatment failure. So this is where we are at the moment. At the top is the process you have to go through in developing the new medicine. You start with design, and Ros did this back in the mid-2000s. Once you've got an idea of what you want your drug to look like, you then have to discover it, i.e. make it and prove that that works. And that's what Ros did up to 2016 when she became very unwell. We're now doing what's called the preclinical development work, and that is to demonstrate that it's safety, the molecule is going to be safe when offered to patients, and put together the mountain of paperwork that you have to have before you're allowed to do a clinical trial. We're well towards the end of that, and no issues have been encountered. The next stage will be the clinical trials and, of course, ultimately marketing. So if we look at the next steps, completion of the preclinical development, as I've said, so far with no toxicity or safety issues. We are going to be confirming the activity of CF10 in a new rat model of cystinosis that has been developed by Jennifer Hollywood and Alan Davidson in Auckland, New Zealand. The models that CF10 has been studied in so far, a mouse model and a zebrafish model, are important and interesting, and the data are encouraging, but there is a feeling in the field that these models are not ideal, whereas the newer rat models that have been developed do seem to be more accurate in modeling the clinical disease. And Jennifer and Alan have demonstrated that cysteamine does work in their model, but have encountered issues which we hope that CF10 will overcome. And we're very grateful to the Cystinosis Foundation UK for supporting this study. And we're just in the process now of shipping the materials out to Auckland, which COVID is making challenging, but never mind, we will get there. The next thing that we hope to start next year is a pilot run-in study with cysteamine in cystinosis patient volunteers. This will be conducted with Graham Lipkin in Birmingham. We're currently developing the clinical protocol for this. And the reason for doing this study is we want to work out the logistics of all of the assays that we will go on to use with CF10 when we're ready to start the CF10 trials, so that there is absolutely no delay. The moment CF10 becomes available, we will be able to offer it to patients and to start the clinical evaluation. Then finally, the CF10 clinical trials themselves, that we anticipate, all going very well, might start in 2023. What we've got to do between now and then is, first of all, to make the CF10 itself in bulk using the required standard of production, which is something called good manufacturing practice synthesis. We then have to formulate it in a way that will be acceptable to patients. These will probably not be the final formulation. This will just be the formulation that will support the initial clinical trials, and then we need to undertake those clinical trials. To do all of that, we are of course going to need funding, and so actually the first next step is to apply for that clinical funding to undertake the work in due course. This is the CF10 team at the University of Sunderland, people who had previously worked with Ros, and then Sally Birtles, who has come in now as the project manager. Colleagues at Birmingham, as I've said, Graham Lipkin will be the PI on the new, initially the run-in study, and then subsequently on the CF10 study. Sundara Pharmaceuticals is a spin-out company that's been developed by the University of Sunderland as required by the Medical Research Council as part of the funding. And then other colleagues, in particular at LabCorp, who were Covance, who are doing all the safety and toxicology studies, and then at High Force Research, who have generated all of the material that we're now using. And finally, just to note the collaborators, co-workers, and the people who have generously funded this work. Of course, the discovery at Sunderland, Rosie's colleagues, Lisa Frost and Paul Hambleton, and then superb collaboration that Ros had from colleagues in the cystinosis research community. Eleanor, of course, is here today, and we now have Jennifer Hollywood and Alan Davidson in Auckland. And the funding from Cystinosis Foundation has been truly outstanding. I know Ros was very, very keen that this project should be taken forward, given this very significant funding that she received from CF UK, and I hope CF UK are reassured that that's going to be the case.